virology or plant virology. So we can see what will be the probability questions that we can expect in ASRB net 2023 plant virology. So before that, let's start with the definition of plant virus. So actually, the virus is a submicroscopic, infectious, and obligate intracellular parasite. We know very well. So I will tell you some interesting facts about this. The term virus was first coined by M.J. Byjernick. M.J. Byjernick. The meaning of virus is poison or venom or infectious liquid, something like that. So Byjernick is considered or Byjernick is considered as father of plant virology, and he also coined some other terms like contagium vivum fluidum, because the infectious liquid of mosaic disease of tobacco, he named that infectious liquid as contagium vivum fluidum. 2021 this question was asked. Contagium vivum fluidum was coined by Byjernick, something like that. There might be a chances. And if you see the general properties of plant viruses, hope you guys know about all these general properties, but let me go through a glimpse. Okay, this virus contains either DNA or RNA as a genetic material, but not both. Virus does not contain both the DNA and RNA as a genetic material, but it contains only one type of genetic material, maybe DNA or RNA, neither DNA or RNA, something like that, but not both. Okay, most of the plant pathogenic virus genome contains RNA as a genetic material. Around 70 to 78% of the plant viruses contains RNA as a genetic material. That too, positive single-stranded RNA. So here generally the symptoms of viruses, if you see, yellowing is the quite common symptom, because once the plant enters into the plant cells, it stops the or it inhibits the production of chlorophyll. If there is no chlorophyll, automatically that leaves turns into yellow in color. So where the virus moves from cell to cell through plasmodesmata. So from inside the cells, the inside the plant, the virus moves from cell to cell through plasmodesmata. So various kinds of symptoms that the viruses can produce, like mosaics, or leaf curl, circling, stunting, necrosis, vein clearing, vein banding, inhalation, various kind of symptoms that the viruses will produce. So most of the viruses can transmit from plant to plant through insect vectors. Around 60 to 70% of the plant viral diseases are alone transmitted by aphids and leafhoppers, around 60 to 70%. Okay, and normally virus can replicate by using an enzyme called RNA polymerase 1. It's a very important. They can ask like this: which enzyme is responsible for the replication of plant viruses? RNA polymerase 1. Whereas RNA polymerase 2 is meant for viroid. RNA polymerase 1 for virus, whereas RNA polymerase 2 for viroid. So this is some general description about plant virus. So we'll see the suffixes. So order ends with virales or virales, or family ends with viridae, subfamily ends with virinae, and genus ends with virus. This is quite common for, if you take any virus, this is quite common. So let's see the important plant viruses and their genome, genetic material. So in exams they will ask some, there might be a chance, and I have seen many times. So maize-streak virus genome is composed of, or Gemini virus genome is composed of, or something like that. They will ask these genome structures. So here starts with the single-stranded DNA. So all Gemini virus, banana bunchy top, that is BBTV, banana bunchy top virus, it is transmitted by a insect vector called black aphid, that is Pentalonia nigronervosa. And the next one is the maize streak, beet curly top, bean golden mosaic. All these viruses whose genome is composed of single-stranded DNA. Second one is a double-stranded DNA, cauliflower mosaic virus, rice tungro bacilliform virus, banana streak virus, and sugarcane bacilliform virus. These all viruses whose genome is composed of double-stranded DNA. Then single-stranded RNA, tobacco mosaic virus, tobacco necrosis virus, tomato spotted wilt virus, potato mop top virus, turnip yellow mosaic virus. All these are transmitted by, whose genome is composed of single-stranded RNA. The last one is double-stranded RNA, wound tumor virus, lettuce bigweed virus, rice ragged stunt virus, rice dwarf virus. The virus genome is composed of double-stranded RNA. There are plenty of other viruses also there, but I just mentioned some important plant viruses. Definitely we may expect like banana streak virus genome is composed of, or which of the following virus, they will give virus name like beet curly top virus genome is composed of single-stranded DNA, double-stranded DNA, or single-stranded RNA, double-stranded RNA, something like that they will ask. So we may expect something like this. So let's have a general knowledge or general description about virus, virusoid, viroid, and all those things. So let's talk about virus. I think I already told you in the first slide itself. So now I will tell you some other important facts about virus. So when it comes to the shapes of virus, there are different kinds of shapes are there, like rigid, rod, isometric, or basiliform virus. Sometimes like these kinds of viruses are there, right? So the first reported viroid in India is sugarcane mosaic virus. Sugarcane mosaic virus is the first reported virus in India, that is reported by J.F. Daster. J.F. Daster. So virus, the name virus was coined by Bijernich. So in the year of 1898. So the first reported virus, many viruses are there, but when it comes to the first properly reported virus is tobacco mosaic virus. The two are given by Bijernich only. And the first Indian reported virus is sugarcane mosaic virus, that was reported by J.F. Daster. So let's have something about virusoid. So virusoid was the first, sorry, not virusoid, sorry, viroid. Okay, so viroid was first reported by Diener and Reimer. Diener, D-I-E-N-E-R, Diener and Reimer in 1971. Year is also important, definitely they will ask. Year is also important. And the first reported viroid is potato spindle tuber viroid. That is the first reported plant viroid. By Diener and Reimer. So the difference between virus and viroid is virus contains nucleic acid and outside protein coat, that protein coat which protects the virus. So when it comes to viroid, it is also called as virus devoid of protein coat. It doesn't contain any protein coat. That is the main difference between virus and viroid. Viroid is devoid of protein coat or absence of protein coat that we can observe in viroid. Okay, let me tell you, first reported viroid is potato spindle tuber viroid, and it was first reported by Diener and Reimer. So when it comes to the definition, we can also expect something like this. Which of the following pathogen is virus devoid of protein coat, or which of the following pathogen doesn't contains any cellular, what can I say, that protein coat or something like that, they will ask. That is viroid, Diener and Reimer. So I will tell you some interesting facts about. So actually the genome size ranges from, the viroid genome size ranges from 246 to 401 nucleotides. The smallest viroid is coconut kadang viroid, is the smallest viroid, which contains 246 nucleotides. 246 nucleotides. And as I told you, virus is replicated by using RNA polymerase 1 enzyme, whereas viroid is replicated by RNA polymerase 2 enzyme. So most of the viroids are transmitted by SAP transmission, but so far there is no vector has been reported that involved in the transmission. So far 40 viroid diseases are reported. The first reported viroid is potato spindle tuber viroid, and the smallest viroid is coconut kadang viroid, which contains 246 nucleotides. I think I have given the nucleotides and all those things in some other slide that I will tell you. So the next one is virusoid. So when it comes to virusoid, it is also like a circular single-stranded RNA, but it is dependent on plant viruses for replication and encapsulation, whereas virus and viroid never dependent on anyone, but virusoid dependent on viruses for replication and encapsulation. It was first discovered by Randles in 1981. So this is some general description about this. And when it comes to the next one, prions. It was the word prion was actually first given by Stanley Prusiner. The word first given by Stanley Prusiner in 1982. So coming to the definition, Normally prions are, like a very small infectious proteins, I can say infectious or folded proteins, where it contains nucleic acid, but where the infection in nature will spread from one protein to another protein. So it is a very small infectious protein where it can able to spread their nature, or it can able to spread their infectious nature from one protein to another protein. This is prions. Very important thing that I would like to tell, prions is otherwise called as slow viruses. Slow viruses are otherwise called as fuzzling protein. Prions is otherwise called as slow viruses. Prions is otherwise called as slow viruses because it kills the host very slowly, and also called as puzzling protein because the protein is the infectious protein, right? The protein is the infectious protein, and it can able to spread their infectious nature to other non-infected proteins. And the next one is satellite virus. I'm telling you, I'm giving you the general description. Hope you guys understand, because we got very less time to deal all those things. So if we got more amount of time, so that we can discuss in detail about each and every single topic, so we're also planning to do that. Definitely we will do. And the next one is the satellite virus. Satellite virus was first reported by Casanis in 1962. The first reported satellite virus is tobacco necrosis satellite virus, that is TNV or TNSV, we can say tobacco necrosis satellite virus. So these are all the, when it comes to the definition, so the satellite viruses are actually subviral agents or nucleic acid that is enclosed by a protein coat. So it doesn't contains any necessary genes for reproduction, necessary genes for replication, so that it need to dependent on another virus, that is a helper virus, for replication, multiplication and co-infection. That's why it called as satellite viruses. It is also like a viruses, but due to lack of necessary genes, it need to dependent on helper virus for replication, multiplication, as well as co-infection. Okay. And the smallest satellite virus so far discovered is a tobacco necrosis satellite virus, that is 17 nanometer. I have already separately placed this about these sizes and all those things. There we will discuss. The second one is mycoviruses. Myco means fungi, viruses means viruses. Mycoviruses are the viruses or mycophages, the virus which infects and replicate inside the fungal cell. So mycoviruses means virus which infect the fungi. Virus which infect the fungi is called mycoviruses. Here the genome of mycovirus, it's very important. I have seen this question. The genome of mycoviruses is composed of double-stranded RNA, and single-stranded RNA is also be reported. When it comes to roughly 70% of the mycoviruses whose genome is contains double-stranded RNA, and the remaining 30% is contains single-stranded RNA. You just remember it is a double-stranded RNA, okay? And double-stranded RNA. So this is about mycoviruses. Actually, mycoviruses was first reported by Hollings. Mycoviruses was first reported by Hollings. So the next one is cyanophases. Cyanophases. Actually, the virus parasitizing the cyanobacteria, that is called cyanophases. Mycoviruses means virus which infects the fungi is called mycoviruses, whereas cyanophases means viruses which infect the blue-green algae is called cyanophases. It was first discovered by Safferman and Morris. It is a very important. I have seen this question. Safferman, Safferman, Safferman and Morris in 1963. Safferman and Morris 1963. And cyanophages are actually classified like LPP1, that is starts with their algal genera name Lingba, Plectonema and Formedium. So that is LPP1 named. So these are all about general like virus, viroid. You just remember what is the first virus, who discovered it, something like that. Virus was first discovered by Bijanac in 1898. Let me recap once again, a little bit fast. So virus was first discovered by Bijanac in 1898. The first virus is tobacco mosaic virus. Viroid, Diner and Draver 1971, and protospindle tuber viroid. Virusoid. So virusoid was Randless 1981, and prions infectious proteins, that is Stanley Prusner. Satellite virus Casanis 1962, that is first one is tobacco necrosis satellite virus. Gemini virus. Oh, we haven't discussed about Gemini viruses, right? So anyway, let me come. So we discussed about mycoviruses. Mycoviruses are the viruses which infect the fungi is called mycoviruses. Cyanophagous. Mycoviruses were discovered by Holding. Cyanophagous means virus that infected the blue-green algae or cyanobacteria is called cyanophagous. Let's discuss about the Gemini viruses. Gemini means twin particle. Very simple. Gemini means twin particle. You remember something that Gemini virus whose genome is composed of single-stranded DNA. It's a very important. I have seen this question before. And Gemini viruses. Gemini means twin particle. Gemini means twins. Okay. Gemini viruses genome is composed of single-stranded DNA. So for replication, co-infection, everything, it needs a twin particle, double particle. That's why it's called as Gemini viruses. So now we'll discuss about phytoplasma. So before entering into phytoplasma, before entering into the various kinds of disease that is transmitted, so let's discuss what is phytoplasma. Phyto means plant, right? So now I will tell you the phytoplasma was first discovered by Doe et al. In 1967. 1967, that is infected with mulberry plants. So far various kinds of plant viral diseases have been discovered so far. So the first reported phytoplasmal disease is mulberry dwarf, which is transmitted by leaf hopper, and the leaf hopper scientific name is Ischimonus setellus or Ischimonus picetus also. You can also call it as picetus, okay? Ischimonus stellatus. Now we'll see, let's have a general look about phytoplasma. Actually, phytoplasma was first discovered by Doe et al. In 1967, that is infected in the mulberry plants. So they thought that it is some kind of maybe virus or something like that. Later, they identified it as a phytoplasma. Before it is called as a mycoplasma-like organism. Now it's changed to phytoplasma. So let's come to the definition. So actually, it's a wall-less. Wall-less means there is no cell wall. Instead of cell wall, it contains like a membrane, triploblastic membrane. So here the important thing is the genome of phytoplasma and spiroplasma. Both the phytoplasma and spiroplasma contains both DNA and RNA, and also contains ribosomes also, okay? Phytoplasma and spiroplasma contains both DNA and RNA as a genetic material. Actually, this phytoplasma belongs to the order, I think, Achiloplasmatales, genus normally Phytoplasma. Why it is called candidatus phytoplasma? Because candidatus means cannot cultured. So that's why it's called as candidatus phytoplasma. So as I told you that it contains both the DNA and RNA as a genetic material. The actual genome size ranges from 530 to 1350 kb. Here I mentioned a few phytoplasmal disease and insect vector and their transmission. It's very important. Little leaf of brinjal, it's a very important one. Little leaf of brinjal is transmitted by leaf hopper. And one more thing I would like to tell you, most of the phytoplasma and spiroplasmal diseases are transmitted through leaf hopper. You can see out of 10 diseases that I have given here, 7 or 8 was transmitted by leaf hopper. So leaf hopper is the major insect vector involved in the transmission of phytoplasmal diseases. So little leaf of brinjal is transmitted by insect vector leaf hopper. The scientific name is Ischimonus picetus or Amarasca bigutula. And the next one is sesame pillodi. It is also very important one. Sesame pillodi, it is transmitted by leaf hopper. That is Voracious albicinctus. And grassy shoot of sugarcane, which is transmitted by leaf hopper, that is Saccharosydnea saccharivora. And one more aphid also involved here, Rafflesia maedis. And coconut lethally living transmitted by plant hopper, that is Mindus crudus or Hapalaxia crudus. Rice dwarf transmitted by leaf hopper, Nephrotitis virescens. Mulberry dwarf, which is transmitted by leaf hopper, it is transmitted by Ischimonus stellatus. European stone fruit cellosis transmitted by psyllids, that is Cocosella pruni. And peach X disease is transmitted by leaf hopper, that is Collodonus and Scapetopius. And pear decline, pear scilla, which is transmitted by Scilla pyricola. And tomato big bird, which is important. Tomato big bird is transmitted by leaf hopper, Uroscius argentatus. Sandal spike, it is also very important. Sandal spike is a which of the following pathogen is responsible for sandal spike, something like that they will ask. Leaf hopper, transmitted by leaf hopper, that is Jasus indicus. And apple proliferation, it is also a phytoplasmal disease, but vegetative propagation is also one of the responsible, one of the reason for behind this. So these are all the important vectors involved in the transmission of important diseases and their transmitted vectors. We may expect one. So the next one is spiroplasma. Uh, I think we already before discussed. I told you about pytoplasma, right? Now is pyroplasma. Actually, pytoplasma was first discovered by Dewey et al. In 1967, whereas pyroplasma was first discovered by Davis et al. Davis, Davis et al. In 1972. 1972. Okay, and Davis et al. 1972. So the definition is. It is also like pytoplasma is a wall-less helical motile prokaryotic organism that is actually bounded by a triploblastic membrane. It's belonging to the genus Mycoplasmatales and the order Mycoplasmatales, genus Mycoplasma, that is pyroplasma. And in here the spiroplasma in cultures are like that can appear like a fried egg or poached egg appearance in the culture. In the culture, the fried egg or poached egg appearance we can observe. And first spiroplasma can be cultured in like Saligo. Saligo is a scientist who first cultured spiroplasma, that is spiroplasma citri, for the first time. Actually, here I mentioned a few spiroplasmal diseases, but the first reported of spiroplasmal disease is constant, which is transmitted by leafhopper, Dalbulus maedis, or Spiroplasma kunkeli. Okay, and citrus stubborn is transmitted by leafhopper. As I told you, most of the phytoplasmal and spiroplasmal diseases are transmitted by insect vector leafhoppers. You can see here most of the diseases. Okay, citrus stubborn is transmitted by leafhopper, that is Circuliferous tenalis or Scapetopius nitridus, and constant is transmitted by leafhopper, that is Dalbulus maedis or Spiroplasma kunkeli. The next one is periwinklelosis. It is also transmitted by leafhopper, that is Spiroplasma phoeniceum. So these are all the important spiroplasmal disease and their insect vectors involved in transmission. So let's have a simple recap about this virus, viroid, pytoplasma and spiroplasma. So virus was first discovered by Bijernek, and the first reported viroid is tobacco mosaic virus, and the year of discovery is 1898. So viroid was first discovered by Diner and Raymond. The first discovered viroid is potato spindle tuber viroid, and the year of discovery is 1971. So pytoplasma was first discovered by Doe et al. And the first discovered disease is mulberry dwarf, and the year of discovery is 1967. Spiroplasma was first discovered by Davis et al. The first discovered spiroplasmal disease is constant. The discovery year is 1972. We may expect maybe one or two questions, something like that. So if we study like this, it will be very easy for us to remember or understand, right? Nucleic acid composition and protein composition, protein code compositions of different plant viruses. So we'll see what are the different kinds of variations are there. So in general, all viruses contains 5 to 40% of nucleic acid, 60 to 95% of protein code. In general, I'm telling, okay. In general, almost all the viruses, whatever the virus you can take, all the viruses will fall, most of the viruses will fall the nucleic acid content between 5 to 40, as well as protein 60 to 95. Hope you guys know virus contains only two things: nucleic acid, that is the infectious material, and the protein code, which covers or which protects the nucleic acid that is inside. So almost all viruses contains only two things: nucleic acid and protein code, that spikes and other things, that's secondary. So most of the viruses contains nucleic acid and protein code, right? So here we are seeing just composition, how much amount of nucleic acid will be present, or how much amount of protein code is composed of. In general, all viruses contains 5 to 40% nucleic acid, 60 to 95% protein code. Okay, tobacco mosaic virus, which contains 5% nucleic acid and 95% protein code. Turnip yellow mosaic virus, which contains 34% nucleic acid and 66% protein code. Potato virus X and potato virus Y both contain 6% nucleic acid and 94% protein code. Cowpea mosaic virus, which contains 31 to 33% nucleic acid, 67 to 69% protein code. Cucumber mosaic virus contains 18% nucleic acid and 82% of protein code. Tobacco ring spot virus, which contains 40% nucleic acid and 60% protein code. These are all the nucleic acid and protein compositions of some important plant viruses. Okay, so let's discuss about tobacco mosaic virus alone. It's a very important virus, since it's been almost 125 years since the discovery of tobacco mosaic virus. Still this tobacco mosaic virus have importance. It's still it's serving as like a model system for many of the experiments. It's been almost 125 years since the discovery. I will tell you one small interesting fact about this. In 2011, this is the scenario of 2011. Out of 7,000 publications are available on virology, 6,000 publications are alone published on tobacco mosaic virus. Now see how important the tobacco mosaic virus. It's like a model pathogen. Everything, whatever the experiment we want to conduct, so many discoveries is also related to this tobacco mosaic virus. Now I will tell you what are the different kinds of discoveries and how important it is. So let's have some general description and general knowledge about this tobacco mosaic virus. Tobacco mosaic virus actually belonging to the genus Tobamovirus. Now Tobamovirus. Now actually this Tobamovirus comes under the family Virgaviridae. Virgaviridae. Genus Tobamovirus, family Virgaviridae. We discussed that it contains, the genome is composed of RNA, that is positive single stranded RNA. Sometimes this question also we can expect, like tobacco mosaic virus genome is composed of positive single stranded RNA. So actually it is composed of 5% nucleic acid and 95% protein coat. I think we have seen this in a previous slide. Let's tell something. So tobacco mosaic virus contains 158 amino acids, 2130 or 2130 capsomere subunits or protein subunits, and 6400 nucleotides, or if I want to tell you exactly, 6395. So around roundly we can round up, we can call it as 6400. So in exams we can expect questions like this: tobacco mosaic virus belonging to the genus, or tobacco mosaic virus genome is composed of, what is the protein content, protein coat content or percentage of tobacco mosaic virus, how many amino acids are there in tobacco mosaic virus, what is the capsomere, number of capsomere subunits of tobacco mosaic virus, or number of nucleotides of tobacco mosaic virus, something like that we can expect. So actually this amino acid, complete amino acid sequence of tobacco mosaic virus was first done by Anderer and Suguta in 1960. I repeat, complete nucleotide sequence was first done and reported. Tobacco mosaic virus contains 158 nucleotides. After complete nucleotide sequence only, Anderer and Suguta confirmed in 1960. And 2130 capsomere subunit, 6400 nucleotides. And the tobacco mosaic virus contains 130 helix ton subunits. So if you see the picture of tobacco mosaic virus, it will be like banana bunch. It will be like a banana bunch. Inside protein, inside nucleic acid will be there, and this all banana bunch are protein subunits. Okay. Then it contains 130 helix ton units, and that was rounded up by 1 by 3. If we calculate exactly, we will get around 2130 capsomere subunits, something like that. So tobacco mosaic virus molecular weight is 17,600 daltons, or 17.6 kilodalton, or 17.6 kd, or 17.06 k. That was determined by Knight. He is the scientist who actually determined the molecular weight of tobacco mosaic virus, Knight. So the genome size of tobacco mosaic virus is around 6.4 kb. I told you that 6400 nucleotides. So the genome size is around 6.4 kb, and the refraction index is 1.57 is the refraction index. So TMV genome can be altered. It's a very, very important question. I have seen this question. The genome of TNB can be altered. So we can alter the genome by using ADP, adenosine diphosphate, ADP. Okay, ADP means adenosine triphosphate. So by using ADP, we can change, or we can alter the genome of tobacco mosaic virus. The molecular weight is around 39 million Dalton. The nucleic acid molecular weight of tobacco mosaic virus is 2 into 10 to the power of 6 Dalton. So actually tobacco mosaic virus is a rigid helical rod-shaped particle. It will look like a rod, like this. Rod-shaped particle, where their measurement size is 300 into 18 nanometer. 300 is the length, 18 is the width, something like that. So 300 into 18. So the thermal inactivation point. So if you want to kill tobacco mosaic virus, we need to hit up to 93 degrees centigrade. Most of the plant viruses can be killed at the range of 55 to 70 degrees centigrade, but TMV can withstand up to 93 degrees centigrade. So the thermal inactivation point of TMV is 93. Dilution endpoint is 10 to the power of 6, and TMV producing inclusion bodies. Inclusion bodies are simply like, what can I say, that these are all the some moles of viruses, like a simply indication structures. Okay, so to prevent the continuous production of viruses or immediate killing of cells, the viruses produce these kinds of inclusion bodies. So inclusion bodies are also used for the identification of plant viruses, because each and every plant virus produce different kinds and shapes of inclusion bodies, whereas TMV produce inclusion bodies is called X bodies, which are crystalline and anamorphic inclusion bodies. And TMV resistant gene is N1. N1 is the resistant gene of TMV. And cell-to-cell movement of tobacco mosaic virus is mediated by plasmodesmata. Plasmodesmata. Here, actually, this plasmodesmata is actually smaller than that of tobacco mosaic virus. Imagine this plasmodesmata. Imagine this is plasmodesmata. This is virus actually, right? So this big virus cannot go through plasmodesmata because the size of the plasmodesmata is actually smaller than that of virus. So how this TMV is mediating or going from cell to cell through plasmodesmata? Because the virus can produce a protein called movement protein. It produces a protein called movement protein. So with the help of this movement protein only, TMV can transfer from one cell to another cell. It's like a shortcut here. Okay, it's like simply like a shortcut from cell to cell movement of TMV is mediated by, or the virus can move from one cell to another cell through plasmodesmata. Here the plasmodesmata is smaller, but still the virus is going because the production of a protein called movement protein. So this movement protein helps like it mediates in transfer of a virus. So the TMV produce a movement protein of 30 kilodalton, and that is P30. And TMV is not actually transmitted by insects or nematode or any other vectors, because most of the plant viruses, I told you in the first slide, most of the plant viruses are transmitted by insect vectors. But when it comes to TMV, it is not transmitted by insect, not transmitted by nematodes, or not transmitted by any other vectors, but it transmitted by mechanical transmission or sap transmission. So mechanical transmission is sap transmission is the method of spread or mode of spread. So TMV contains 4 ORF ring. ORF means open reading frame. So 4 ORF rings. Okay, and a few other things that I would like to tell you is the tobacco mosaic virus produce a movement protein size is 30 kilodalton. I told you, na? Coat protein size is 17.5 or 17.6 kilodalton, and a replicase enzyme size is 126 kilodalton and 183 kilodalton. As I told you how important the plant virus is, now I will tell you so that you guys will come to know how important the TMV is. Though, so the first recognized plant virus in the world is TMV. The first inclusion bodies produced by the plant virus to be noticed. The first inclusion bodies produced plant virus to be noticed is also TMV, that was reported by Ivanovsky. And the first cross production technique was developed by McKinney in 1929 in TMV. We thought that actually cross production technique we studied extensive and citrus tristeza virus. Now we are employing extensive and citrus tristeza virus, but it is actually discovered, it is actually meant for or developed for tobacco mosaic virus. So tobacco mosaic virus, and later scientists identified tobacco, rather than tobacco mosaic virus, the cross production technique is very effective against citrus tristeza virus. So now that's why we are employing this cross production technique in citrus. So the first identified phenotypic mutant of plant virus is also reported in tobacco mosaic virus, that is also reported by McKinney. The first virus to be chemically purified or crystallized, we know very well. The first purification of plant viruses was done by W.M. Stanley in tobacco mosaic virus. So it is the first virus to be chemically purified. It is the first virus to be chemically purified by Stanley in 1935. And when it comes to the first coat protein to be sequenced, that is the amino acids. Okay, the complete amino acid, I repeat, complete amino acid sequence was first published by Anderer and Suguta, and they reported tobacco mosaic virus contains around 151 to 158. They told that 151 to 158, and finally they come to a conclusion: the tobacco mosaic virus contains 158 amino acids. The next one is antigen. So first antigen was actually observed or developed from tobacco mosaic virus. First antigen actually developed for, and sap transmission. So sap transmission, the first reported sap transmitted virus is also tobacco mosaic virus. Coat protein mediated resistance was actually also actually employed and developed on Paul Abel and B. Chettle 1986 and tobacco mosaic virus only. And the first virus to encode cell-to-cell movement protein, that is a movement protein. I told you, na, plasmodesmat is actually smaller, but again, still, even though it's a smaller, the tobacco mosaic virus is managed to spread from one cell to another cell through plasmodesmata. It's a plasmodesmata is simply like a shortcut. Okay, so by producing a movement protein, it is it is mediating its transfer. So first proved specific antigen infecting plants is also reported in tobacco mosaic virus. First serological studies of plant virus is also reported in tobacco mosaic virus. See how important the tobacco mosaic virus. That's why I just telling some important information about tobacco mosaic virus. So now we'll see the difference between viroid. As I told you that viroid, we already discussed about viroid. In viroids, there are two groups are there: pospiviridae and avsunviroidae. So in pospiviridae, CCR is present. Pospiviridae, CCR is present. They will ask something like the questions like CCR is present in which of the following group of viroids? Pospiviridae, avsunviroidae, something like that. CCR means central conserved region. Central conserved region. So central conserved region is present in pospiviridae, whereas CCR is absent in avsunviroidae. And ribosome activity present in pospiviridae, whereas it is absent in avsunviroidae. The shape of a molecule is pospiviridae circular. It is also circular. Secondary structure is a rod-like, whereas here in avsunviroidae it's a branch. So the replication mechanism is asymmetrical rolling circulation. It's a very important asymmetrical or asymmetrical rolling circle mechanism, whereas here is symmetrical rolling circle mechanism. The site of replication is a nucleus in pospiviridae, whereas site of replication in avsunviroidae is chloroplast. So number of rolling circles is only one rolling circle in pospiviridae, two rolling circles in avsunviroidae. So the examples, what are all the different kinds of viroids that comes under is potato spindle tuber viroid and coconut lethal yellowing, coconut kadang I told you that is the smallest viroid reported so far. So coconut kadang potato spindle tuber, so both are comes under pospiviridae. And avocado sunblotch, chrysanthemum chlorotic mottle viroid, that comes under avsunviroidae. The important things that we need to consider is CCR. CCR is present in pospiviridae, absent in avsunviroidae. And the second one is the asymmetrical rolling circle mechanism can be seen in pospiviridae, whereas symmetrical can be seen in avsunviroidae. One rolling circle, two rolling circle. And the next one is site of replication of pospiviridae is nucleus, whereas chloroplast where avsunviroidae takes place. So this is some general information about avsunviroidae and pospiviridae. So these are all the different kinds of viroids and their groups. As I told you, avsunviroidae, in avsunviroidae, polymoviridae, along something like that, avsunviroidae and pospiviridae. So avocado sunblotch, chrysanthemum chlorotic mottle viroid. So this is the largest viroid. Okay, chrysanthemum chlorotic mottle viroid, which contains around 390 to 401 nucleotides. 400. This is the largest plant viroid. Patent leech. Peach latent mosaic virus, that is contained 355 to 339. So avocado sunblotch is also contains smallest one. It is also smallest one, 246 to 251. But but coconut is the kadang is the definitely smallest one. So I have given so many things here. Let's discuss only important things. Okay. The most important thing we need to consider is that is chrysanthemum chlorotic mottle viroid. That is the largest viroid, which contains 398 to 401 nucleotides. That is the one important one. Second one is citrus exocartosis. Exocartosis contains around 368 to 375. I would say 375. And then the next one is potato spindle tuber viroid. That is the first reported viroid, right? So potato spindle tuber viroid contains around 356, 357 or 3 up to 361 it contains. Okay, then coconut kadang, as I told you, right? Coconut kadang contains 246 to 247, and it is the smallest viroid that is reported so far. And coconut tinangaja contains 254. Coconut tinangaja contains 254, and these are all some important plant viroids and all those things, because we can't say plant viroids because virus, so far we know very well that virus can infect both animals and plants, but viroids can only infect plants, and there is no viroid has been reported so far that can infect humans or animals. Viroids are especially meant for plants only. Okay, Some important plant viruses sizes we will see. So as I told you in the previous slides that important plant viruses, smallest plant size of sizes of plant viruses. So the first one is the satellite tobacco necrosis virus, or tobacco necrosis satellite virus is the smallest virus that is reported so far, 17 nanometer. The second one is the tobacco necrosis virus, that is 26 nanometer. See here, tobacco necrosis satellite virus. Okay. The next one is the tobacco necrosis virus, that is a little bit different. And bromo mosaic virus, that is 28 nanometer. Copy mosaic virus 28 nanometer. Tobacco ring spot virus 28 nanometer. Cucumber mosaic virus 30 nanometer. Vica virus 30 nanometer. Tomato bushy stunt virus 30 to 35 nanometer. Alpha mosaic virus 30 to 50 nanometer. And this is important, cauliflower mosaic virus, the size of cauliflower mosaic virus 50 to 52 nanometer. 50 to 52 nanometer. And lettuce necrotic virus 60, potato leaf roll virus 90 nanometer, Rio virus 65 to 70, and wound tumor virus 70 nanometer. It's also very important. And the sugarcane mosaic virus 11 into 750 nanometer, and bean common mosaic virus 12 into 750, plumppox virus 12 into 760, Badna virus 300 into 20 nanometer, tomato spotted wilt virus 100 nanometer, and it is not straight, it is spherical in shape. Okay. So common indicator plants that we can use in virology, now we will see virology studies. For general, the virus, for virus identification or virus indication, goose foot is used as indicator plant. Goose foot. Scientific name is Chinapodium amaranticolor. Citrus greening can be identified by using sweet orange as an indicator plant. Scientific name of sweet orange is Citrus sinensis. Citrus exocortosis, trifoliate orange. Scientific name is Citrus trifoliate. Citrus tristiza, Clagsy lime or Mexican lime. Citrus aurantifolia, protospindle tuber viroid, tomato used as indicator plant, Solanum lagobascum or Solanum bertholdi, and red rot of tea, fish poison bean, that is Tephrosia vogeli, and Phytoplasma periwinkle used as indicator plant, the Catharanthus roseus. So these are all the different kinds of indicator plants along with their used in their plant viral. It's a very important. And previously I have seen many questions from this, like like Citrus tristiza virus can be identified by, or Clagsy lime used as an indicator plant for, or sweet orange used as indicator plant for, something like that we can definitely expect. Then inclusion bodies that is produced by the plant viruses. So as I told you previously that each and every virus produce different kinds of inclusion bodies. Now we'll see. So tobacco mosaic virus produce X bodies as inclusion bodies. Cauliflower mosaic virus produce amorphous inclusion bodies. Vica virus produce granular inclusion bodies. Rhabdovirus produce bullet shaped inclusion bodies. Poti virus produce pinwheel inclusion bodies or cylindrical inclusion bodies. It's very important, like pinwheel inclusion bodies is produced by, or bullet shaped inclusion bodies is produced by, something like that we may expect. Now transmission of plant viruses through various vectors. So as I told you that most of the plant viruses is transmitted through vectors, right? Now we will see what are the various types of vectors involved in the transmission. So tobacco mosaic virus, cauliflower mosaic virus, potato virus X, tomato mosaic virus, cucumber mosaic virus, all are primarily transmitted by sap transmission. Few of the virus that I reported here is also can be transmitted by other means of transmission, but majorly transmitted by sap transmission. Tobacco mosaic virus, cucumber mosaic virus, potato virus X, tomato mosaic virus, cucumber mosaic virus, or cauliflower mosaic virus, all these are transmitted by sap transmission or mechanical transmission. As I mentioned that some of these viruses also transmitted by other vectors, also something like that. The next one is a cucumber mosaic virus. Cucumber mosaic virus is also transmitted by aphids, Apis carnivora and Apis gracile. That I told you what are all the, here all the viruses that I mentioned here are transmitted by aphids. Papaya ring spot virus, sugarcane mosaic virus, potato leaf roll virus, peanut stripe virus, bean common mosaic virus, beet yellow virus, potato virus SY, groundnut rosette virus, peanut virus, alpha mosaic virus, cauliflower mosaic virus. These all are transmitted by aphids also. On the right side they have given the scientific name of the insect vector. P. anation is transmitted by P. aphid, that is Acanthosporium pasum, and the cauliflower mosaic virus having a bimodial type of transmission. So as I told you that it is known to transmit by sap as well as aphid. So two vectors involved, that is called bimodial. And citrus stridza virus is a very important transmitted by brown citrus aphid or black citrus aphid, Toxopteros citrusida and Apis gassofi. And banana bunch-top virus and cutter disease of cardamom, very important. Banana bunch-top virus and cutter disease of cardamom is transmitted by black aphid, that is Pentalonia nigronervosa. And leaf hopper, so as I told you that this aphids and leaf hoppers, these two are the major culprits, and most of the plant viral diseases are transmitted by these two vectors alone. 60 to 70% of the viral disease, if I want to tell. And rice dwarf virus, rice tungrobacilliform virus, wound tumor virus, maize streak dwarf virus, chlorotic dwarf virus, beet collie-top virus, rice grassy stunt virus, rice ragged stunt virus, papaya bunch-top, not banana bunch-top, papaya bunch-top, and maize streak. These all are transmitted by banana bunch-top is transmitted by black aphid, but papaya bunch-top is actually, it's not a virus, it's like mycoplasma like organism. Okay, fastidious vascular bacteria, sorry, that is FVB. It is transmitted by leaf hopper, that is Emposca papaya and Emposca stevensi. I think I have mentioned this in general plant pathology itself. So these are what are all the viruses that I have mentioned here, all are transmitted by leaf hoppers. So the next one is sandal spike and little leaf of brinjal, Sasamum pillodi, grassy shoot of sugarcane. All are patoplasmal diseases were transmitted by leaf hopper. Coconut lethal yellowing and coconut foliar decay are transmitted by plant hopper. Rice stripe virus is transmitted by brown plant hopper. And tobacco leaf curl virus, cotton leaf curl virus, tomato leaf curl virus, tapioca mosaic virus, mung bean yellow mosaic virus, bendi yellow mosaic virus, cassava mosaic virus, bean golden mosaic virus, African cassava mosaic virus, all are transmitted by white flies. All are transmitted by Bemisia tabaci species. And tomato spotted wilt virus, it is a very important. Tomato spotted wilt virus is transmitted by thrips tabaci, something like that. Groundnut bud necrosis virus also transmitted by thrips palmi. Tobacco streak virus also transmitted by thrips frankiella. Tobacco ring spot virus is transmitted by thrips tabaci. Sunflower necrosis virus is transmitted by thrips Scrotothrips drossolis. These all are transmitted by thrips only. Then squash mosaic virus transmitted by beetles, Diabrotica duodecim punctata. And cowpea mosaic virus is transmitted by beetles. And cocoa shoal shoot virus is very important. Cocoa shoal shoot virus is transmitted by mealybug, Pseudococcus nichalensis. Not only cocoa shoal shoot virus, but also banana streak virus. However, I told you that banana bunch-top virus and cutter disease of cardamom is transmitted by black aphid, Pentalonia nigronervosa. So these two also very important. That is cocoa shoal shoot virus and banana streak virus is transmitted by mealybugs. Scientific name is Pseudococcus nichalensis. Beet collie-top virus transmitted by lacybug. And citrus greening, it's very important. Citrus greening is transmitted by psyllids, Diabrotica citri or Triosa erythrae. Wheat streak mosaic, ryegrass mosaic, peach mosaic, shallot X or PG&P sterility or PG&P sterility mosaic virus is transmitted by mites or areopid mites. Among this, wheat streak mosaic virus, Asaria tulipae, and PG&P sterility mosaic virus are very important. PG&P sterility mosaic virus, otherwise called as red gram sterility mosaic virus, Asaria kajani. It is actually first reported by an Indian scientist, P. Lavakumar. Now he is a very famous scientist, and now he is in Ibadan, Nigeria. He's a very famous scientist he is. And potato virus X, potato mop-top virus, wheat mosaic virus, lettuce big bean virus, beet necrotic yellow bean virus, tobacco necrosis virus, rice stripe mosaic virus, Indian peanut virus, all are transmitted by fungal species. Very important here. Potato virus is transmitted by Syngentium endobiotic, which is a fungi. Potato mop-top virus is transmitted by Spongospora subterranea, it's also a fungi. Wheat mosaic virus is transmitted by Polymyxa graminis, very important. Lettuce big bean virus is transmitted by Olephidium brassicae. Beet necrotic yellow bean virus is transmitted by Polymyxa betae. And Indian peanut clump, I have seen this question many areas. Rice stripe and Indian peanut clump virus, both are transmitted by fungal species Polymyxa graminis. And ring spot virus, aphid mosaic, prunus necrotic ring spot, very important. Prunus necrotic ring spot virus is transmitted by pollen. It is also known to transmit by seeds, but majorly it is transmitted by pollen. Okay, prunus necrotic ring spot, prunus necrotic ring spot is transmitted by pollen. And the next one is the grape fan leaf virus is transmitted by nematodes, Ziphiniuma index. These are also very important. Grape fan leaf virus is transmitted by nematode, Ziphiniuma index. Tobacco rattle virus is transmitted by Trichodorus and Paratrichodorus. Tobacco ring spot virus is transmitted by Ziphiniuma. P. early browning mosaic virus is transmitted by Trichodroid. So these are all some important concepts and things that we can expect from virology section. Hope it will be useful. Stay geeky, stay tuned. We are Team Geeky Researchers.